So, yesterday's lecture did not record on Panopto for some reason. IT's looking at it, so I'm going to make a recording now and go through the slides again so you have this. So we talked about yesterday action potential, so we were talking about nervous tissue. We started off with resting membrane potential here at minus 70. We said that was maintained by potassium leakage. So the action of potassium leaking out of the cell and sodium potassium ATPase pumping it back in keeps this membrane potential stable at minus 70. So we went through the different types of cells that there are, the oligodendrocytes, which make myelin inside the central nervous system. So they can myelinate multiple axons in the central nervous system. So myelin was a lipoprotein. It's made out of fat and protein. So it is a good insulator. It doesn't conduct electricity. It doesn't allow charges to cross. So myelinating these axons prevents any current leakage as an electrical signal is traveling through them. Plus it prevents short circuiting, so axons don't come into contact with each other, and thus a signal can't jump from one axon to another. Schwann cells are in the peripheral nervous system, and they basically wrap around an axon as a single cell. So there are multiple Schwann cells myelating this axon. So Schwann cells are only associated with one axon, whereas oligodendrocytes in the CNS are associated with multiple axons. Astrocytes form the blood-brain barrier. They've got these foot pads that wrap around capillaries that carry blood through the brain. And so the capillary endothelium, so the cells that make up the capillary, those simple squamous epithelial cells, plus the foot plates of the astrocyte forms a blood-brain barrier. So that prevents things from getting into the CNS. And neurons are postmitotic. They do not divide and replace themselves. They don't undergo mitosis. So once you kill them, they're gone forever. So the blood-brain barrier prevents damage to the central nervous system. We said we were going to use the multipolar neuron as our model as we were going through this, and then over here is where we would find the dendrites. Right. They attach to the cell body, the soma, which is basically this, and then the soma grows an axon. In that area that you can see in the microscope where the soma becomes the axon, where the axon grows off of it, it's referred to as the axon hillock. Now somewhere in this region is going to be the first segment where you will find voltage-gated sodium channels. So that's going to be the initial segment. And when an action potential is produced, it's going to be called the trigger zone. The trigger zone, the definition is basically the first membrane segment that contains voltage-gated sodium channels. Right, so a depolarizing event anywhere over here gets transmitted towards the initial segment, and if it's above threshold, then those voltage-gated sodium channels open and you get an action potential. Other types of neurons that we talked about were unipolar cells, which are found in invertebrates. We won't talk about those. Bipolar cells we won't talk about either. Those are in the special sensory systems like the eye. Sooty unipolar cells are associated with sensory systems, touch, temperature, pain. We will talk about those when we do sensory physiology. And there are different types of multipolar cells. We won't be going into pyramidal or Purkinje fibers. We'll be focusing mostly on motor neurons. We said that axonal transport is necessary for substances that are made in the cell body to be transported down in the anterograde direction towards the synapse. And then because we're balancing exocytosis and endocytosis, the things that come in via endocytosis may travel up to the cell body in a retrograde fashion. Motor proteins transport these. Kinesin is your anterograde motor protein, and dynein is your retrograde motor protein. We also discussed the fact that myosin and actin interact. Myosin is a motor protein that transports things within the cell as well, but it attaches to microfilaments, which are made of actin. So establishment of the resting membrane potential is a separation of charges. And the separation of charges ends up such that there's going to be a bunch of positive charges on the outside or extracellular side of the membrane, and they're attracted to the negative internal environment of the cell, the ICF. And then on the inside of the cell, there are a bunch of negatively charged ions close to the cytoplasmic side of the membrane. They're attracted to the positive ions that are on the outside of the cell. So this separation of charges creates essentially a battery. You've got a positively charged pole and a negatively charged pole. And as long as they are separated, as long as they don't come into contact with each other, just like a battery, you've got stored potential energy. As soon as there is some way for them to come into contact with one another, though, then you will get charges moving, and that's called an electrical current. And the way we usually do this is by opening and closing channels. Channels may be non-specific. They may allow both or multiple different types of ions to travel through them. The channels that we're going to talk about mostly are specific. So, for example, voltage-gated sodium channels primarily allow only sodium to travel through the channel. So other ions like chloride and whatever can't travel through that channel. So the action potential is generated when there's a depolarizing stimulus. So if I draw a little multipolar neuron here, give it an axon and a synapse. All right, so up here there might be another synaptic connection with another neuron. So a neurotransmitter gets released at this synapse that causes depolarization right here in this segment. That depolarization is referred to as a graded potential. It's graded because it can be various sizes. You can have small depolarizations, large depolarizations, etc. If the depolarizing event reaches threshold, then voltage-gated sodium channels open. Threshold is basically the change in membrane potential you need to open voltage-gated sodium channels. When that happens, then sodium comes rushing into the cell, and that's referred to as an action potential. Action potentials don't have different sizes or different intensities like graded potentials do. Action potentials are all the same size. Every action potential is an opening of all of this voltage-gated sodium channels. They all have the same threshold value. this influx of sodium into the cell, which causes the cell to rapidly depolarize up to about plus 30 millivolts. And then while the sodium channels are open, potassium channels will start to open. They have their own threshold. Their threshold's right about here. So as sodium is coming in, the membrane charge is becoming less negative, and so then the potassium channels will open. But they're slower than sodium channels, so it takes them a while to open and it takes them a while to close. At this value of about plus 30, the sodium channels will close. And then the potassium that's leaving the cell now is causing the cell to repolarize. Right, so potassium rushing out of the cell causes repolarization. Because potassium channels are slow, that repolarization actually goes past resting membrane potential. So there's a brief period of hyperpolarization. And then, because of this action of the sodium-potassium ATPase, when the potassium channels close, there's a reestablishment of the sodium and potassium gradient, and you get a slow creep back up to resting membrane potential. Two refractory periods we talked about. The absolute refractory period is basically due to sodium channels. We said there are two gates on sodium channels, so if I draw a sodium channel here, right, so there's one gate which is an activation gate, and one gate down here which is a deactivation gate. So under normal conditions over here, before we reach threshold, right, the activation gate is closed. The activation gate opens at threshold, and now sodium can flow in through this open channel. Up at this plus 30 millivolts, the deactivation gate closes. And that's what's being represented here by this little ball. So that sodium channel, even though the activation gate is open, it can no longer allow sodium to travel through the channel because the deactivation gate is closed. You see here the deactivation gate stays closed for a while. And that's what gives you your absolute refractory period. So what has to happen is the cell has to get back down to resting membrane potential. So the activating gate closes, and after the activation gate closes, the deactivation gate opens up. So until sodium channels are reset back to their original position, as they are over here, the cell cannot generate another action potential. Those sodium channels cannot open until they're reset. And that's referred to as the absolute refractory period. At no time during the absolute refractory period can another action potential be generated because the sodium channels can't open. Now, once the channel is back to the activation gate being closed and the deactivation gate being open, now it can generate another action potential. So at this point, if there was another depolarizing stimulus which got to threshold, then you could get an action potential. So beyond that point, until the resting membrane potential, which is now hyperpolarized at the end of the absolute refractory period, until that gets back up to normal resting membrane potential, you're in a relative refractory period. During the relative refractory period, you need a higher than normal stimulus to generate an action potential, but you can generate an action potential. You can see this graph here that talks about excitability, shows you that there's zero excitability during the absolute refractory period, so you cannot generate an action potential. And then as the relative refractory period progresses, so as this resting membrane potential slowly creeps back up to normal, the excitability of the neuron increases until you're back to normal resting membrane potential at the end of the relative refractory period. Now you're back to having a highly excitable neuron. So remember that action potentials are all or none. There's no big action potential or small action potential. They're all the same size. Absolute refractory period is caused by sodium channels not being able to open, so you cannot generate an action potential during that period. Relative refractory period is caused by a period of hyperpolarization caused by slow closing of the potassium channels. So you get too much potassium leaking out of the cell, and the cell becomes more negative than normal resting membrane potential. Let's talk about this positive feedback nature of sodium channels. Sodium channels opening up cause sodium channels in an adjacent segment of membrane to open up, which cause sodium channels in another segment of membrane to open up. So this action potential regenerates itself all the way down the axon, and it's the same at every segment. Doesn't matter. You can stick a recording electrode into every segment of the axon and measure action potentials, and they're all going to be exactly the same. As far as the potassium channels are concerned, they're generating negative feedback because they're repolarizing the membrane. So when they repolarize the membrane, they cause sodium channels to close. So that gives you a negative feedback that turns off the action potential. Now, propagation along the axon, if it's not myelinated, basically you get an activated segment. And you get a wave of sodium that comes in. Right, sodium travels in both directions. It activates the next segment of membrane, causing voltage-gated sodium channels there to open. Again, you get sodium coming in both directions, but because the absolute refractory period, right, when this next segment starts generating an action potential, the previous segment is in the absolute refractory period. So the action potential, this wave of sodium, keeps traveling down the axon towards the axon terminus or the synapse. In myelinated axons, you don't have to worry about that. It's a little faster because the active node is in between the gaps in the myelin. Right, so this is myelin here, and here, and here. And so basically, the action potential is jumping from node to node. Anatomically, those are called nodes of Ranvier. You can see them in the microscope. That's where you'll find clusters of voltage-gated sodium channels. And this jumping from node to node is referred to as saltatory conduction. And it's more rapid than conduction in unmyelinated axons because there's no current leakage. and you don't have to activate every single patch of membrane along the way. You only have to activate the nodes. So if you think about if you're trying to pass something from seat to seat across the room, right, there's a delay every time you hand it off to the next person. Then he has to turn and hand it off to the next person. Then she has to turn and hand it off to the next person. So by the time you get from point A to point B, it has taken longer than if the person sitting at the beginning had just thrown the object and the person at point B had caught it. So conduction in unmyelinated axons is slow, and conduction in myelinated axons is fast. Now, because the resting membrane potential is established by sodium and also the negatively charged organic anions inside the cell, mostly proteins, which are not found outside the cell, the resting membrane potential is highly susceptible to changes in extracellular fluid potassium concentration. So when blood potassium is in normal range, right, so 3.5 to 5.0 milliequivalents per liter, then you have what's called normokalemia and a subthreshold stimulus. A stimulus, a depolarization, a graded potential that does not reach threshold, does not open voltage-gated sodium channels, and you do not get an action potential. But a threshold or suprathreshold stimulus, once it reaches action threshold value, voltage-gated sodium channels open, and you do get an action potential. Now, changes in ECF concentration of potassium are going to affect the resting membrane potential. More potassium in the extracellular fluid, what is called hyperkalemia, causes the resting membrane potential to be closer to threshold. So the neuron or the muscle cell or whatever cell it is becomes hyperexcitable. It's more easy to generate an action potential because the resting membrane potential is closer to threshold. So this subthreshold stimulus, which would not generate an action potential under normokalemic conditions, under hyperkalemic conditions, it may generate an action potential. So you're going to end up in hyperkalemia with things like muscle spasms, maybe even tetanus, because of this increased excitability of these cells. Hypokalemia, less than or lower ECF potassium, moves the resting membrane potential farther away from threshold. So it makes it more difficult to stimulate this electrically excitable cell. So back here under normokalemic conditions, a stimulus that would reach threshold and cause an action potential may be a subthreshold stimulus in hypokalemic conditions. So you're going to have muscle weakness, you're going to have an inability of muscles to contract. Under hypokalemic conditions. Now the heart is a muscle, so hyperkalemic conditions can cause it to contract too rapidly, possibly even contract and not relax. Hypokalemic conditions would make it more difficult for the heart to contract. So you would get fewer heartbeats, and they wouldn't be as strong. So changes in ECF potassium can be devastating to multiple organ systems, particularly the nervous and muscular systems, the electrically excitable systems. We classify neurons based upon size primarily, and that size is the diameter of the axon. So the largest fibers are grouped as type A fibers. Intermediate fibers are type B, and the smallest fibers are type C. And then we break down those into even smaller, even more specific classifications. So A alpha fibers are the largest, and they're well myelinated. So up here, if I say this has a lot of myelin, and this has no myelin, so the degree of myelination increases as the fiber size gets larger. So A alpha fibers are motor neurons, alpha motor neurons that control skeletal muscle fibers. They're the largest diameter fibers, so they conduct action potentials the fastest. So they have the highest conduction velocity in meters per second. A beta fibers are a little smaller than A alpha fibers. They're still fairly well myelinated. They're for touch and pressure, but because they're smaller in diameter, they have a smaller or a lower conduction velocity than the alpha motor neurons. The gamma fibers, those are gamma motor neurons, and they send information to muscle spindles. We'll talk about muscle spindles later. Just make sure that muscle contractions are smooth and not jerky. They're smaller, and of course they're going to have then a smaller conduction velocity. A delta fibers are pain and temperature fibers. We'll talk about those. Again, they're smaller than the alpha, beta, or gamma fibers, and so they're going to have a conduction velocity that is less. The gamma and delta fibers are pretty close to one another. There's actually a little bit of overlap. The B fibers are preganglionic autonomic neurons. Those are smaller than all types of the alpha fibers. And so they're going to have an even slower conduction velocity. Fibers that transmit a slower pain signal, what's called slow pain, are type C fibers. Those are relatively unmyelinated and have a slow conduction velocity, as do postganglionic sympathetic C fibers. I talked about banging your elbow, how that mechanical stimulus can trigger an action potential in the axons that are traveling through the ulnar nerve. So in that case, the trigger zone actually ends up being a node of Ranvier, whichever part of the axon gets compressed. So that's a pressure stimulus. And the fibers that are most susceptible to pressure stimuli are the largest fibers, the A fibers, which is why then when you bang your elbow, the thing you're going to get as a stimulus is going to be that tingling. You may get some pain, right? You're not going to affect the B or C fibers unless it's a really, really hard hit. So if it's a really good whack, you could end up with lingering pain, which is caused by stimulation of those C fibers. Now, hypoxia, or reduction in tissue oxygen, stimulates the B or affects B fibers' ability to conduct an action potential before it affects A and C. Now, the interesting thing, at least for pharmacists, is local anesthetics, mostly of the cane class, Novocaine, benzocaine, lidocaine, things like that. They hit the smaller C fibers first. Right? The purpose of a local anesthetic is to block pain transmission. Right? So Novocaine is given whenever you get some oral or dental work done, and that's supposed to block the pain stimulus that comes through. It does it by blocking voltage-gated sodium channels. So if voltage-gated sodium channels can't open, these neurons can't fire action potentials, so there's no painful stimulus coming into the central nervous system. Usually, though, dentists will give you a whopping dose of Novocaine. Some dentists are very good. They'll give you a little bit at a time till they dull the pain. Most dentists find it easier to just kind of give you a pretty good dose of Novocaine. And that will also affect the A fibers, the larger fibers. So these touch and pressure fibers, which will give you numbness, right? You won't be able to feel not just pain, but also touch. And it'll also affect alpha motor neurons that are traveling through that same nerve that are going to muscles. And so you'll get a droopy lip, maybe your tongue, one side of it will be paralyzed. You'll be talking up funny, right, until the Novocaine wears off. Sensory neurons, we have a different classification system. We call them type 1, type 2, type 3, type 4 fibers. That equates to the A fiber, B fiber, C fiber designation. So the 1A fibers are A alpha. The 1B fibers are also A alpha. So the type 1 fibers equate to the A alpha grouping. Type 2, A beta. Type 3, A delta. That's going to give you your fast pain and your temperature, some of your cold receptors. And then type 4 sensory neurons are going to be your slow pain fibers.